Hello, and welcome to another in our series of NCBI resources webinars. I'm Peter Cooper from the NCBI User Services Group. Today, I'm going to show you how to use the Variation Viewer, a relatively new tool at the NCBI that's a dedicated browser for human genomic variation. Here's what I'm planning to cover today. I'll first mention several of our variation resources at the NCBI, and then I'll use the Variation Viewer to do a number of things. We'll look at small and large-scale variants. We'll look at how to set sequence viewer tracks and options. We'll download results displayed in the viewer. We'll see how to upload your own variants to see their genomic context. Then I'll show you how to link to other variation resources and related databases here at NCBI. So going through some introductory slides, I'm going to do a live demonstration that focuses on one of the desmocollin genes, DSC2, on chromosome 18. This gene has variants that are causative for right ventricular cardiomyopathy, and this is a condition that can cause you to suddenly drop dead. So these variants are among those that genetic testing labs are required to report, even if found incidentally. Let's begin with an overview of important NCBI variation data sources. The resources listed on this slide are resources at NCBI that we typically call databases. They're more than that, since they have their own specialized displays, search features, and associated analyses. Many of them are part of the Entree system that you may be familiar with. These are the sources of the markers and other information that will show up one way or another in the demonstration that we do today. The first resource listed is dbSNP, which is also called just plain SNP in the Entree system. This is the repository of small-scale variations, which are mostly single nucleotide variants. dbSNP also contains repeats or smaller insertion-deletion variants, typically less than 50 bases. What you're going to see in the variation viewer are the reference variants, or RefSNPs, the non-redundant set of variants from dbSNP. dbVar is a repository for large-scale variants. These are insertions, deletions, and copy number variations, often covering several genes or large portions of a chromosome. Within that database, you're going to see things called variant calls. These are the individual reports of a variant. Then there are variant regions that are a non-redundant representation of variable regions in the genome that are based on these calls. Unlike dbSNP, there's no concept of a reference variant. ClinVar is a database that has information about phenotype assertions for both large and small scale variants. These variant phenotype assertions are submitted from a number of sources, including literature only, such as OMIM, clinical testing labs, research labs, locus-specific databases, expert panels, and professional societies. Lastly, I want to mention dbGAP, the database of genotypes and phenotypes, which contains genotype and phenotype association studies. These include results from molecular diagnostic assays, case control studies, longitudinal cohort studies like the Framingham study. And one thing to know about dbGAP is that access to the individual level data requires formal permission that we call controlled access. There are some public data analyses available. There are also specialized tools and browsers associated with NCBI variation resources. 4 of these are listed here. The first tool I want to mention is the Variation Reporter, which allows you to map your variants onto the data in dbSNP and onto the genome itself, and to predict the functional consequences of those variants. I'll not have time to show you how to use that today, but it's a useful entry point to the variation viewer since the results are displayed in the viewer itself. Another important tool is the 1000 Genomes browser, which has sequence alignments and variant calls from the 1000 Genomes project data. We'll see some of the minor allele frequencies today from the 1000 Genomes project in the variation viewer. Clinical Remap is another tool. This is used for mapping variants onto RefSeq gene records. RefSeq gene records are typically specially curated sequence records with a fixed coordinate system. These are made for over 5,000 human genes at this point. These include many of biomedical interest. For example, the DSC2 gene that we're going to use today has a RefSeq gene record. Finally, we have the Phenotype-Genotype Integrator, or PhGene, which has phenotype-genotype association data from a number of sources, including dbGAP, NHGRI, Genome-Wide Association Studies Catalog. So keep in mind that the results that you get here are typically associations. They're not necessarily causative for a phenotype, but genetically linked to it. We'll see some of these association results in the variation viewer today when I do the demonstration. All these tools are linked from various displays at NCBI, but I also want to point out that there's a tools page, and that's the URL at the bottom of this slide that you can link to directly that has a list of these tools. Another separate page that I want to show you is something called the variation portal page. This exists outside the Entrée system at NCBI, but it's useful because it offers one-stop access to all the NCBI variation resources, including the tools. For instance, you can see the link to the variation viewer right here in the center of this slide, as well as the other tools that we've already mentioned and the variation databases. In fact, when I do the demo, I'm going to get to the variation viewer directly from this page. The URL for this page is listed at the bottom of this slide, and it's pretty simple to remember. It's just the NCBI homepage URL, www.ncbi.nlm.nih.gov, followed by the word variation. There are 3 allied resources that I want to mention. These are resources that are really about human health, and they complement the standard set of variation resources at NCBI. First one is MedGen. MedGen is an aggregator of information on human disorders and other phenotypes. MedGen provides a vocabulary for describing and classifying disorders and syndromes. Another resource related to MedGen that's more familiar to many people is Online Mendelian Inheritance in Man, or OMEM, which is now maintained outside of NCBI. OMEM is a compendium of information from the biomedical literature about human phenotypes with a genetic component. There are records for phenotypes, genes, and selected variants in OMEM, and it's a source for many of the records that are in ClinVar. And finally, I want to mention the Genetic Testing Registry, which is a repository of submitted information on genetic tests with information on the purpose, the methodology, validity, and the utility of the test. They also include context for the test and credentials for the testing lab. This is a resource that clinicians and genetic counselors can use to find a genetic test for a specific condition or variant. I'm going to now show a few slides about how to use the variation viewer and some of its features, and then we're going to use these features in a live demonstration. This is a screenshot of the main variation viewer page. In fact, if you go there fresh with no previous internet cookie set in your browser, you're going to see the gene we're looking at here, which is complement factor H on chromosome 1. I want to show you some of the components of the viewer itself from this slide. There are 5 main components that you can use for navigating and finding things in the variation viewer. These are the search feature, the gene and exon navigator, the sequence viewer, the variant filter, and the variant table. I'm going to go over each one of these briefly here, and then we'll see how to use them. Search feature is the main way to enter the viewer and to find a region, gene, or components of interest. You can do lots of different kinds of searches here, and I'll show you how to do some of those. The gene and exon navigator is a handy way of shifting the view or focusing on a specific part of a gene in the viewer. Embedded within the variation viewer is the NCBI sequence viewer itself, and you've probably seen this in other contexts at NCBI, like in gene or in nucleotide. You can add and manipulate the tracks and do lots of other things using this component. The variant table gives you a table of the variants in the region displayed in the viewer. We'll come back to that in more detail in a few minutes. The variant filter is a way of selecting which particular variants are shown in the table. And I also want to point out that you can expand this your data section of the viewer to upload your own data. I'll show you a way to add other tracks as well through the configure dialogues in the sequence viewer in a few minutes. I want to show you a couple of aspects of the search component. You can, of course, simply type terms in the search box here and press that arrow to run the search. You can do lots of different kinds of searches, and the kinds of searches that are useful are given to you as examples and a link there. For example, you can search for a gene symbol, chromosome position, cytogenetic band, accession numbers. There I've got a rough SNP accession number and a nucleotide sequence accession number. You can search for human genome variation society expressions for variants. You can also search for phenotypes, and that's what we're going to do in the demonstration to do today, is to look for a phenotype. Here's the Gene and Exon Navigator, another way of getting around. It lets you browse by gene. You can do that using this double-headed arrow here. It will jump you to the next gene in the view. Notice there's one on either side here. The single-headed arrow will let you navigate by exon. You can jump to a specific exon, or you can use the radio button here to pick out the exon that you want to jump to. As I said a few minutes ago, you can upload data into the viewer. This is a really simple VCF, variant call format file that I made. It loads, and when it loads, it produces a track in the sequence viewer section that shows me how my variants map onto the sequence that's displayed. This is the filter component, and these are the various attribute filters that are available. I'm going to go over these in more detail when we use them. I'm not going to read all of them out to you right now. This is a way of just getting the variants that you want displayed in the viewer. This is sort of the main view of the data. This is the variant table. First of all, let me point out that you can download things directly from here, and this will give you all the data that's in the table in an XML format, which is easily parsable. And we're going to look at the links within the table, and what those basically provide you with is a way of integrating these data with the other kinds of databases that are at NCBI. So I'll just point out what some of them do. So if I click through on the dbSNP ID, I can go directly to dbSNP. The link here that has the location will take you into that position, will zoom you into that position in the variation viewer. These are links to sequence ontology terms. These are the things that classify the kind of variant and the molecular consequence, and that will take you to a separate website that explains that vocabulary. The link here under clinical significance, and that's classified as pathogenic, this particular variant, that takes you to the ClinVar record for this. Notice that there are two sources of minor allele frequencies shown here. One is the 1000 Genomes Project data, and the other is the NHLBI Grand Opportunity Exon Sequencing Project, GoESP it's called. It's another set of populations. Notice that the two populations have slightly different minor allele frequencies. Also, of course, the variant details shows you, which I've expanded here, that's an expandable part of this viewer, it shows you how these particular variants affect the NCBI reference sequences, and here they are, the two splice variants for this particular gene and their corresponding proteins. Let me just show you that there's a link to two very important resources at NCBI, Gene and PubMed, and linking out to these will give you access to lots of other kinds of data that are related at NCBI. And so what I'm really showing you here is the way this kind of viewer integrates with the rest of NCBI. So from these links in the viewer, I can link out to all these variation databases and also to the sort of central databases like Gene and PubMed, and from there I can link out to all kinds of additional data. Before I move on to the demonstration, I want to point out to you some of the help documentation that's available for both the variation viewer and the other variation resources at NCBI. Notice that there's a help document and an FAQ linked directly to the variation viewer. You might have seen that in one of the screenshots at the top. By the way, the URLs on this particular slide are just shortened so that you just put the NCBI homepage URL in front of these to get the complete URL. There's also a viewer video on YouTube. There's a direct link to that YouTube video. It gives you sort of an introduction to using it. We have an education page. A link to that is a fact sheet page. There are a number of fact sheets that are very good introductions to these resources that I've listed for you. Here are the names of the ones that are about variation resources. Also, for general help, I just want to remind you that we do have a help desk. In fact, I'm a member of that help desk. So if you have a question about anything you see in this video or anywhere else at NCBI, you can write to info. That's the address given there near the bottom of the slide. If you have a question about this webinar, its content, or comment about it, or you want to know about webinars in general, you can write to that webinars address at the bottom of the slide. Okay, let me give a brief introduction to the demo that we're about to do. We're basically going to focus on a particular region on chromosome 18 that contains some interesting genes. There's a small family of genes with some pathogenic variants. These are desmosomal proteins. They're the desmocolin genes and desmoglin genes on chromosome 18. Some of these have causative variants for arrhythmogenic right ventricular cardiomyopathy. So we're going to browse around there and look at some things. We're also going to upload data, and we're going to add some tracks. I'm going to add the association results track and the prologous pseudogenes track, and we'll talk about how those are useful. So what I'm going to do now is click on this link here, and that took me directly to the NCBI homepage, which is where I'm going to start with my demo. And I have a lot to show you, so I might not get through all of it because we want to save some time for questions, but we can certainly get some good ideas about how to use this resource. There are lots of ways to get to the variation viewer. Starting from the NCBI homepage, it might not be particularly obvious how to get there, but an easy thing to do is to start from one of the traditional variation resources. You can always get there through the resource list here, and of course you can get there the way a lot of people do by typing variation viewer in Google. I'm going to go to the SNP page here. This is the home page for SNP or dbSNP. And notice that you can go from there to that page that I showed you in the slides. This is a variation portal, which is a pretty good way to get around in the variation resources. And here is the page that has basically links to all the things that we talked about in the introductory slides. And I'm going to go ahead and follow the direct link here to the variation viewer. Okay, and if you come here fresh, that is, you haven't used the variation viewer recently and you don't have a cookie stored, you'll wind up looking at the complement factor H gene on chromosome 1. I'm not going to use that gene as an example, although it would be a good one. I want to just show you how to search a little bit in here, so I'm going to search with the phenotype. Notice that there's a nice little section of the page that you can click on here that will tell you some good example searches, the kinds of things that you can search with in this box and get good results. So what I'm going to do is search for a phenotype, and I'm going to search for the one that we were talking about before. So it's right ventricular cardiomyopathy. And so the way you work this search device here is you click the little arrow after you've entered a term, and you may get lots of results. In this particular case, we do. There are lots of genes here, and many of them are involved in the desmosomal complex there. The gene that I've chosen to work with is DSC2, which is on chromosome 18, not chromosome 19, chromosome 18. And if I go ahead and follow the little blue arrow that appears when I select that line in my results here, that will load that gene. Okay. So now I have some interesting looking data here in the viewer. Let's just go over some of the aspects of the viewer itself. Now we're going to get our way down here eventually to looking at these variants in the variant table, which is really the meat of the whole thing. That's what we're trying to do. But at the top you can see there is an ideogram representing chromosome 18 with the various cytobands labeled. And you may notice that you're at a particular place here on the Q arm, the Q12 band of chromosome 18, and you're zoomed in looking at the DSC2 gene graphic here. So let me just go over some of the other tracks that are here. So we're going to skip over this navigator here. We're going to come back to that in a few minutes, and then I'm down in here looking basically at the graphical sequence viewer. The first track at the top is the representation of chromosome 18, so that's the sequence track. It's the one track that's not labeled in the viewer, and it shows you the coordinate system here. The next track that's available is gene track, and you can see there's a graphic representing the gene. This is a merged graphic that represents all the different splice variants. The sort of vertical spots here represent the exons, the horizontal lines represent the introns, and the arrow tells you the direction of transcription. Notice that in this particular case, this gene is on the opposite strand of the one that's represented for chromosome 18, so it's pointing sort of the other way, if you will. The next track is the short variations that are in ClinVar that are from dbSNP. The track below that is dbSNP itself. So you'll notice that the dbSNP track at this zoom level is basically just continuous data. The ClinVar short variations, these little boxes are approximately where those variants are. And the colors tell you something about how pathogenic they are, whether there are any pathogenic variants in there or not. So if you ever don't understand what's being represented on one of these sequence viewer tracks, you can just mouse over the top of the track there and click on the track legend, and it'll show you what that particular legend is.
In this particular case, it tells you about what SNPs in there are pathogenic. So if it's purple, it means at least one SNP in that bin has been marked probable pathogenic. Okay, let's go ahead and show you some of the other tracks here. The last one here, of course, I've mentioned the DB SNP track. And then finally, there's a ClinVar large variation. So these are variant regions from DB var, where there are copy number variations. They're going to be sort of either deletions or duplications. And again, if you wanted to see what that track legend was, you could click here to see the track legend. I'll tell you that the red ones are deletions and the blue ones represent duplications. A couple of things to do from here. One of the things to do, first of all, is to just another sort of sequence viewer kind of thing to do. I'm going to mouse over the gene track here at the object that represents the gene. And let's just take a trip out and we'll go to gene itself just so we can get some biological context for this particular example. So I'm going to gene, this is for desmocolin 2 for human, and it talks about the fact that there's a little family of cadherins on chromosome 18, the desmocolins and the desmoglines, and it mentions the fact that we talked about this, that mutations in this gene are associated with this right ventricular dysplasia and other kinds of cardiomyopathies. So let's go back to the variation viewer. One more thing I want to do here before we leave is modify the track setting here for gene. So I'm going to go up here to the gene track again, and we're actually going to display the configuration dialog box for the graphical sequence viewer. And I'm going to go in here and modify the track settings. What I want to try to do is to demonstrate the different splice variants for this. And those will become important when we try to navigate across the exons for this particular gene. So I'm now looking at the configure dialog box, and what I want to do here is go ahead and show all, which is going to expand that gene graphic to show me everything that's there, including the transcripts themselves and the proteins. And I'm also going to project the SNPs from MRA and CDS feature, and we'll see what that does in a few minutes once I configure this. Okay, so now if we look at the gene graphic here, it shows us the gene more clearly. And you can see the green object there represents the gene itself. The blue objects represent the transcripts. The red objects represent the proteins. And you can see that there are SNPs actually mapped onto them. And we'll take a look at that in a few minutes. Let's just use the Gene Exon Navigator to sort of investigate these a little bit more. Notice that now I can see in the Gene Exon Navigator at the top the 3 different splice variants. There are 2 NM type reference sequences that are aligned here. There's also an XM that's based on next-gen sequencing data. I'm going to pick the second transcript here and pay attention on the right-hand side to the Gene Exon Navigator. What you'll notice is that it will actually change because I'm going to add an exon when I do that. So you'll notice it was a little subtle, but actually it added an exon. And that's, you may not be able to see it very clearly, but if you look at that second transcript, there's actually a small exon near the 3' end. And you can remember this is on the opposite strand, so the 3' end is in the right-hand end of this view. So if I want to, I can zoom in and look at that by selecting this particular radio button here that represents that exon. And so I've zoomed in, and I'm looking at that exon, and here I have a slightly different protein sequence for this isoform. And notice you can see that the SNPs are mapped onto the variant, the variants are mapped onto the transcript and the protein in that particular case. Now, if I want to get out of this and go back to seeing the whole gene again, how do I do that? I can go up here to the region and notice that I can select the gene. Again, with pad, if I want to, then that's the way it shows by default with 10% padding. So we're back out looking at it as we saw it a few minutes ago. Okay, let's go over and then look at some of the controls here on the left-hand side of the viewer. And what we're going to do here is ultimately get down to looking at the SNPs at the bottom. A couple of things to notice while we're going there. Here's our search feature. We've already talked about that. We're going to, in a moment, upload a file. I'm going to skip over this for a minute just to go ahead down here and talk about GRC issues. Before I do that, and I should have done this before we went down there, notice at the top that you're capable of picking a different assembly. We're now looking at GRCH38. This is the Genome Reference Consortium Human 38 build. This is the current build, and it has NCBI's annotation on it, which is build 106. Notice that I can pick the Genome Reference Consortium build 37 if I want to, which is the previous build with the previous annotation. And many people are still working with that previous build, so it's very useful to be able to see those coordinates here if you want to. Okay, so the Genome Reference Consortium, of course, is a multi-center consortium that's responsible for maintaining and updating the human genome, and they'll build the genome sequence every so often. In between builds, they correct problems that they know about with the genome. So what I'm pointing out to you here in the region details is that there is one GRC issue in this region, and this will be the first time we're adding a track to the viewer. So what I want to do over here is add that track. And so here is the extent of a particular problem that the Genome Reference Consortium is trying to resolve. I'm actually going to show you another thing you can do with this. You can drag the tracks around if you want to. I'm going to pull it down so it's in the position where the sequence track is. If I do it right, it'll work. There it is. Now it's below the sequence track. And if I put my mouse pointer over there, over that object, you can see what the particular issue is. There is a problem with one of the back clones. It seems to be missing a TTA somewhere in the back clone, and so the GRC is working to resolve that particular problem. So we've just added a track to the viewer, and we'll see two other ways to do that during this demonstration. Okay, since we're in this region, I'll show you another way to add something there. And that's this little device here that says your data. If you have your own VCF file, you can upload that here. And I have a small one on my desktop. It's very simple. And let me go ahead and choose that file. Windows thinks that's a card file, but that's okay. I'm going to open that up. And you can see basically what it's reading from my file here. These are the variations that it's reading, and I've placed them on chromosome 18. And here's where they line up to the sequence that's in the viewer. And I can move this track if I want. So if I would like to have it closer to the actual SNP track, I can do that here. And I can see where some of these may line up with known SNPs. Some of them may be things that we've discovered and nobody knows about. So let's move down. Now we're going to move down to the filters down here, and then we're going to look at some variants in the variant table. To make the demonstration a little bit easier, we'll just focus on variants from dbSNP for the rest of this talk. So what I'm going to do is go ahead and select that right now. So that eliminates my large-scale variants from the table, but there's still 893 of them here. And notice that there are lots of things that I can filter by. I can filter by whether they're in ClinVar, what their clinical significance is, the variant types, sort of these sequence ontology terms. I can filter by minor allele frequency, which is kind of interesting. So let's look and see if there are any common SNPs in here. And so I can, by clicking this greater than or equal to 0.05 minor allele frequency, and you can see in parentheses that I'm going to wind up with 2 of those. So I'll go ahead and retrieve those. I'll filter my table basically. So I'm going to wind up with only 2 variants. And they're both reasonably common variants. Notice the minor allele frequencies are here for the 1000 Genomes Project data and for the GoESP data. The variant frequencies are similar but not identical. The populations are different. And you can see the sequence ontology terms here. This is a missense variant, and so it's actually going to change the amino acid sequence of the protein. What I want to do here is to show you that you can zoom in to find that particular variant by clicking on this location in the graphical sequence viewer, in the variation viewer here. And so I've zoomed in. You can see that there are actually 3 SNPs here. This one is actually at a different place in the codon there than the other 2. Now the other 2, the people in dbSNP tell me they realize that these are identical and they will be merged. These are the reference SNPs, and so they'll be merged to make a single record. They represent exactly the same change. And if you look at the codon here, the codon, remember, we're on the opposite strand, so it's ATC, reading this way. Notice that the variations have been mapped onto the objects in the viewer, so you can see the variants on the nucleotide sequences. And then when there are variations on the protein, of course, those are affecting the entire codon, and so you can see them there. And you can see that there are actually two of them. The one allele changes the last position of the codon, makes it a methionine codon, and the one that we're looking at changes the isoleucine to a valine. And I can go ahead and change the region again if I want to go back to the gene with PAD to get back out to where we were before. And then let's do a different kind of filtering here. So I'm going to go back and uncheck the minor allele frequency filter. And then I'm going to ask to see only those variants that are in ClinVar. There are 57 of them. And this might be an interesting set for me in my particular research interest. So I just want to point out to you that I can download whatever's in this table by clicking the download variation button here. And it's going to download this in XML format. You can open that, of course, with a text editor, or you can parse it with some kind of an XML reader, or you can just open it with a text editor if you want to. And it contains all the information that's in this table, including that which you would get if you expanded each one of the entries. Okay, let's go ahead and filter this even further. So I'd like to look at those variants that are in ClinVar that are pathogenic. So I'm going to check that. And there are 3 of them. And they're all going to affect, they're going to have some kind of deleterious consequence because they're all labeled as pathogenic. There are 2 of them that are frame shifts variations and one of them that's a splice acceptor variant. So all those are going to have a bad effect on the corresponding transcript and its protein. I'll go ahead and expand. I'm going to look at the second one in the table. This is this SNP accession that ends in 4041. And you can see that what that does when I expand it is it tells me about what the molecular consequence for each one of the transcript variants. The ultimate thing that happens is threonine 477 is replaced with a frame shift. There's a deletion there, and so you get a methionine there, but then it changes the rest of the protein and probably doesn't get expressed or it doesn't get processed properly. And it gives you the information from ClinVar that it's associated with arrhythmogenic right ventricular cardiomyopathy type 11, and it's pathogenic. So I'll go ahead and follow the link to ClinVar from here. Notice I can do that from the parent here of the thing that I expanded. It's all going to go to the same place. And this is the ClinVar record. And actually a lot of this information has already been shown to you in the table that's there on the variation viewer. Look down here, you can see the details of the clinical assertion. And this is one of those variants that's coming from OMEM. So it says classified by a single submitter, literature only. And from here, if you wanted to, you could link to OMEM or to our own database about phenotypes MedGen. And I'm going to do that from this record. So I could go from here at the bottom. It's also linked up here under clinical significance. There's a link to MedGen here. So I'm going to go ahead and go over to MedGen. And this is the vocabulary describing a disease or syndrome. This is the arrhythmogenic right ventricular cardiomyopathy type 11. And MedGen is a sort of an aggregator, and it also applies various kinds of control vocabularies to these terms that describe diseases. And it pulls in some articles. This one's from Gene Reviews. This is from the Genetics Home Reference, and of course you can read about this one. This is a bad thing. You could have this and not know that you have it and suddenly drop dead. So this is one of those ones, by the way, I'm going to scroll down to the bottom of the record. This is one of those genes that if they find some incidental findings when they do exome sequencing, the American College of it's medical genetics and genomics. That's what's hard. It's got an extra word in there. So this is one of the ones that they recommend that you tell the patient about, even if that's not what you were testing for. And in terms of linking through, this is a good example. You could link through here to PubMed, and I won't show you that. just to save some time. And from PubMed, we actually have the full text article of this, and you could actually look at the table in there that tells you about the particular genes that they recommend that you report. And here you can see the vocabulary that classifies this particular disorder. And you can see here we have links to GTR, our genetic testing registry, in case you were a clinician and you wanted to order some kind of genetic test for this. Okay, so I've got a lot of tabs open, and that usually confuses me, which is not hard to do. So let me go ahead and close a couple of those. And I'm going to go back to the variation viewer. I'll go ahead and restore all this stuff. So a couple of other things that you can do. We're not going to be focusing on large variations, so just to make the display a little bit simpler, I'm going to remove a track. We haven't done that before, so I'm going to go ahead and remove the large variations track. And also I can remove my own track that I made, this chromosome 18 VCF. I'll go ahead and take that away too, again, to make the display simpler. So we're going to add a couple of more tracks to the variation viewer. And I'm going to do that using the configure menu here. And I'm going to focus on variation tracks. And there are two of them that I want to add. One of them is kind of an odd one. It says paralogous pseudogene alignments. This is interesting because you might call a SNP when actually what you're looking at is an alignment to from another paralog or from a pseudogene that has a very similar sequence. So that's something that you might be interested in knowing about. The other thing that I'm going to put in here is the association results. So I'm going to check that box here. And these are associations of variants with particular phenotypes. This is a little bit different than the kind of data that's in ClinVar, because here we're talking about mainly linkage. These things are linked, but they're not necessarily causative. The causative SNP or variant is something that's probably genetically linked or close by. Let me go ahead and configure this. Okay, now you can see that I did add those two tracks there at the bottom of the view here, but there's nothing in this region. So what I'd like to do is to zoom out, and we'll take a little trip over to the right-hand side of my viewer. So we're actually moving down chromosome 18. And there are lots of ways we can do that. And I'm going to do it, first of all, by zooming out a little bit, just so you can see the kinds of genes that are around here. Okay, and you can pick up that little family of the desmocollins here. Here's desmocollin 1, 2, and 3. There is a transcribed gene above it that's a non-coding gene. I can scroll over a little bit more. And notice I'm now leaving the area where there is a GRC issue. And these are the desmoglin genes over here. Actually, I'm going to zoom out a little bit more just so you can sort of see the interesting thing about this particular little set of genes is that this is basically a duplication that's inverted. And so these genes are clearly related to each other, and there's been a couple of different gene duplication events in this region. There's even that non-translated RNA there that's present in this other set. Many of these genes are associated with these kinds of cardiomyopathies, and you can see there are lots of pathogenic variants in the desmoglin region as well. So far, though, we're beginning to pick up a few association results down here. They're kind of color-coded. The redder they are, the hotter they are, the more closely they're associated with a particular phenotype. And here's one that's red. It's on a gene at the end of this. This is a gene called transthyretin. Let me go ahead and zoom in. This is something that we haven't done before. I'm going to use this device here in the sequence range at the top to zoom in. I'm going to zoom on that range. And here we can see that this is an association result, but notice that it's not actually within a gene. It's close to a gene, and that gene is the transthyretin gene. If I mouse over the association result here, you can see that the phenotype that they're measuring here is vitamin A concentration. And this is from the NHGRI GWAS catalog. This is a very strong association, and you can look at the legend just like we did with the other tracks to see that the redder these are, the higher that negative log p-value is. In this case, it's very high, 13.2. So this is a pretty strong association. And this is another one of these cases where, remember, this is not causative. It's just telling you that this particular variant is associated with this particular thing that they measured, which is vitamin A concentration. And if you want to know more about that, of course, what you can do is, what kind of gene is TTR? Well, let me just go to gene real quick. Well, this is actually a carrier protein, and one of the things that it carries in plasma is retinol, vitamin A. So that's a very interesting biological story, that there's something near this gene that affects vitamin A concentrations. And if you wanted to read about that, in the NHGRI GWAS catalog is coming from the biomedical literature, I can go ahead and go here to visit PubMed, and I could read the full text article if I wanted to here. And just to show you, linking back through to the variation resources again, you might notice that if you look in the text up here, there are two different variants mentioned, this one, and they're in different genes. This one they ascribe to this gene called retinol binding protein 4, obviously a very interesting gene if you're talking about vitamin A concentrations. If you want to, you could link through to that, and you could find the cited SNPs. And you'll notice that none of these are associated with retinol binding protein 4 as far as DB-SNP is concerned. I'm just going to click here to show you that I'm now in the DB-SNP record for that one, which was actually listed for a different gene. But if I go back through the chromosome position, it's going to go into the variation viewer, zoomed into that particular SNP. So that's another way into the variation viewer. And that's probably the most common way in, is not starting from nowhere, but linking to it. And I have to zoom out to see something interesting, and that interesting thing is that although it's in the 3' UTR of this other gene, it's also in the 3' UTR of the retinol binding protein 4. So that's particularly an interesting biological connection at any rate. Okay. Last thing I want to do, and we need to stop soon for questions, is I'll go ahead and show you the other track, which is the prologous pseudogene alignments. And what I'll do here is use this device up here to just sort of jump gene by gene. So now I'm moved over, continuing along chromosome 18. And when I use this double-headed arrow in the gene exon navigator, I'm jumping one gene at a time. And eventually I'll come across ones like this one. This is a solute carrier family 25 member, which has lots of alignments to other places in the genome. This is a rather complicated one. I'm going to go and find a slightly more easy example to look at. And I'll leave it to you as an exercise to do this one. But this is a pseudogene. I know it's a pseudogene partly because of the way it's colored in the graphic here. This is a retrotransposed pseudogene. It's for phosphogluconate dehydrogenase. The functional gene is actually on chromosome 1. And here I can see the alignment for chromosome 1 right here. And what you can do, and I'll leave it to you to do this so we can have some time for some questions. I can simply write a query in the search box over there that says to go to that place, this particular range on chromosome 1, and there you will find the functional gene for the gene that we're looking at here, which is a retrotransposed pseudogene copy of it. Okay, so I want to wrap this up here, and I want to go ahead and throw this open to questions. It looks like we have a lot of them based on all the activity that I've seen across the room. So I hope you've entered them into the box before now, because we've probably got a lot of questions built up. If we don't get them answered, we will post the answers along with the rest of the course materials on the webinars page. Okay, Ben, how do you want to do this? Do you want to take over for the questions?
Yes.
Okay.
Well, thank you, Peter. That was a phenomenal webinar. So we've taken some of the frequently asked questions. We won't, like Peter said, we won't get to all of your questions, but we're going to get to as many of the frequently asked ones as we can. So the first sort of general question that was asked, and this is a pretty simple one, but I want to make sure everybody is on the same page, but what are variant calls? So variant calls are when you have a human reference genome, and Peter talked a little bit about what the human reference genome is and how it's made, and then you take somebody's personal genome or exome, you compare it to that, and you believe that you have enough evidence to say that the individual's genome is different in a particular site from the human reference genome. So that is what a variant call is, and there's a lot of caveats to how we do variant calls, so on and so forth, and how the community in general does variant calls. There were some specific questions on this, and we're probably going to try to get to those individually. Some of those questions were very complex, but we'll try to put those on in a sort of public forum as well. So then somebody asked, how do you find what is mutant or wild type in a fairly related and fairly simple question? Sometimes this is controversial, and of course it is. One thing that we commonly use are the minor allele frequencies from 1,000 genomes. So we sequenced about 2,600 individuals from all over the world, and what we're able to see are the frequencies of particular single nucleotide polymorphisms or short nucleotide polymorphisms up to about 10 nucleotides across a whole bunch of ethnic populations across the world. Now I'm going to pass the microphone to Melissa Landrum from the ClinVar project, and she's going to tell you a little bit about the differences between mutant and wild type in ClinVar, and then she's going to move on and answer a couple other questions before she passes the microphone back to me.
So the other approach to finding information about whether an allele is normal or not is to look in ClinVar. So ClinVar is a database of assertions of clinical significance about alleles, and it's important to note that NCBI is not making these assertions ourselves. The assertions of clinical significance are submitted to us. So you can go to ClinVar, look up an allele, and find out what do these clinical labs, research labs, gene-specific databases think about this allele. Do they think it's normal, or does it cause disease? And I would point out that you will not see terms like mutant or wild type in the database. We use more clinically oriented terms. So the wild type alleles would be called, say, benign or likely benign. Mutant alleles would be called pathogenic or likely pathogenic. And then there's also a gray area in between called uncertain significance when people cannot decide, don't have enough information. So the next question is, which ontology is used in the MedGen database? So MedGen, just to remind you, MedGen is really an aggregated database that brings in information from several other databases that have information about diseases and phenotypes. So it brings in information from SNOMED, CT, ULS, HPO, the Human Phenotype Ontology, OMIM. We're currently bringing in data from OrphaNet. So some of these databases do have an ontological structure to them, like the Human Phenotype Ontology. OrphaNet also has its own ontology called ORDO for OrphaNet Disease Ontology. So these are all incorporated into MedGen. You'll also see some manually curated hierarchies of diseases within MedGen, and these are provided by NCBI staff curation. The next question is, do we have information available about heterohomozygosity in Variation Viewer? So the Variation Viewer doesn't have this specific information, but some ClinVar records do have information about zygosity. So if you're interested in an allele, you can click out to ClinVar and see what information is available. The caveats are that the zygosity information is an optional field in ClinVar, so we don't always have it. And also, because this is one of the less frequently submitted fields, it's not available in our web display, so that would be something you would have to get from the XML download. And now I will pass it back over to Ben.
Hi, I would just like to tag on to that question about hets and homozygotes. For those of you who are more advanced users, one way to get at those types of questions would be to get authorized access to dbGaP data, and many dbGaP studies have imputations, or in other words, have phased genotype calls, and so that's somewhere you could get a lot of information about hetero and homozygotes. Additionally, there was a late-breaking question about somatic and germline mutations, and that's another place where you could get that kind of information, although ClinVar does have some information about somatic and germline mutations as well. Another question that was asked, and I think this might be sort of a broad question, is there a template for making your VCF file? As we all know, many of these file types can vary quite a bit. I typically like to use the definition that's on the 1000 Genomes homepage. I think just for VCF files, that has more or less become the industry standard right now. If you want to discuss that offline, please type something into the question module. Another question that was asked, and this may be applicable to many of you, is can you get publication quality images from Variation Viewer? It's a good question. The answer is sort of. So under Tools, you can export a VCF file, or sorry, not a VCF, a PDF file. And so theoretically, you could use that for presentations and possibly even publication. If that's something that you really need to do, something you want to show, go ahead and write a ticket into Help Desk, and we'd love to hear from you about what kind of export formats you'd like to have. Another question that's most likely widely applicable is how do I keep my data in Variation Viewer? And the simple answer to that is get a My NCBI account. So NCBI does not spy on your personal information, but we appreciate it if you sign up for a My NCBI account. This helps you store a whole bunch of types of information, including your NIH BioSketch if you apply for NIH grants, that kind of thing. You can also use it to do sort of frequent and regular literature searches. So we're going to go ahead and sign off. I'd like to thank all of you for attending and paying attention. I know I just said we don't spy on you, but we can actually see whether you're paying attention during the webinar. Once again, thank you for attending, and have a nice day.